And now on to embryonic week 4 again, but now we're looking at neurulation and the neural crest. So we looked at body folding, now we're going back, same time frame, different process. So we'll start with neurulation. So now back at our overview, we're looking at these sections of our overview, which cover neurulation, which is again, differentiation of the brain and spinal cord, and the development of the major regions of the brain. Now we actually have already covered some of neurulation, primary neurulation, in week 3. So we talked about formation of the neural plate, where the ectodermal cells elongate to form the neural plate where it didn't exist before, and the shaping of the neural plate, where the neural plate lengthens longitudinally but remains broader at the cranial end. From there, we pick up in week 4 with bending of the neural plate. So in week 4, we see the formation of the neural folds, which you can see here, and they form at the lateral edges of the neural plate by combining neural epithelium and surface ectoderms. These are combinations of 2 different tissue types, and they rise up dorsally and then ultimately close, closing the neural groove, forming this tube. Now, closure of the neural tube happens when the neural folds adhere to each other. That makes sense. Now, the cells in the neural folds are forming 2 epithelial layers. The roof plate of the neural tube, which is shown in this orangey triangle, and then the surface ectoderm, which would be the future skin. Neural crest cells form in between these 2 layers and migrate throughout the body, and we will talk about them in a bit. Now, closing of the neural tube begins at the occipital and cervical regions and then moves cranially and caudally. So unlike closure of the lateral folds and body folding, the neural tube doesn't start at the far cranial and caudal ends. It actually starts closing sort of in the middle and then proceeds approximately distally in the direction of the head and the tail. Now, the genetics of neurulation are really interesting, but also very complicated and not very well understood. So it's not something that we're going to cover in this class, although of course you are welcome to look at the data that's in the book and to find other sources as well. It's just beyond the scope of what we can do here. Now, in terms of closure of the neural tube and birth defects, there is a major birth defect that arises from failure of closure of the neural tube, and they're called open neural tube defects, which is highly descriptive, more commonly known as spina bifida and anencephaly. These birth defects are essentially the result of holes in the neural tube, results of failure of the folds to adhere, the neural folds to adhere. If the hole is left along the spine, it'll be a spina bifida. If it's at the back of the head, it'll be anencephaly. Anencephaly is lethal because the amniotic fluid can get in and damage the brain, which is non-viable. Spina bifida varies in severity based on the size of the hole and the location of the hole, and part of the module for this week in Canvas is to review the Children's Hospital of Philadelphia website, which covers their care and treatment of babies with spina bifida, both prenatal and postnatal. It's very well done, and I do want you to look at all of the videos and to read the information presented there. All right, secondary neurulation comes after we've actually formed the bulk of the neural tube. So we had talked about forming the neural tube from the neural plate, but now we're going to go down to the caudal end, the tail bud. So within the tail bud, central cells are going to form what's called the medullary cord, and the medullary cord will become a hollow tube and fuse with the caudal end of the neural tube, which completes the neural tube. So in fact, the tail end of the neural tube is derived from different cells than the rest of it. But just like with the rest of the neural tube, the neural crest cells will arise from the roof of this region of the neural tube and migrate. The lateral tail bud cells will differentiate into the caudal somites, and all of this is completed by 8 weeks. Now, the early brain structures are also forming during week 4. Again, we're covering this very briefly, but we will talk about it more in chapter 9. Now, on to our neural crest cells. What are they doing? So, neural crest cells are derived primarily from the neural tube, which are epithelial, but also from these medullary cord cells in the caudal end, or in the tail bud. And they transform into mesenchymal cells and then migrate throughout the body. And this epithelial to mesenchymal transformation is kind of interesting. A, because we have one cell type turning into another, but B, because cancer cells in some cases undergo the same transformation, although of course that's not what's happening in the embryo. And what this diagram is showing you is that the neural crest cells go throughout the body to form a variety of different tissues and organ systems in concert with other tissue types. So what kind of tissue a neural crest cell becomes is based on where it starts. So we sort of subdivide the neural crest cells into regions of origin: cranial, vagal, trunk, sacral, and then the somites are buds of tissue along the spine of the embryo. Cranial neural crest cell derivatives. Now, with the brain, it's even further subdivided because we have sections of the brain at this point. The prosencephalon or forebrain, the mesencephalon or midbrain, and the rhombencephalon or hindbrain. And those are all shown here. Now, the future of neural crest cells does to some degree depend on which section of the brain they're derived from, although in reality, a lot of times they do work together. And there are many, many different cell types and tissue types that are formed at least in part from neural crest cells, and they're listed here. Now, I will say this is a bit of a simplification. So you can see all of these different cells and tissue types derived from cranial neural crest. In reality, they're also derived from other tissue types as well. So it's not as if your teeth are entirely derived from cranial neural crest. Part of them is. I will highlight this one particular derivative, the pharyngeal arches. So the pharyngeal arches are bands of tissue that form sort of at the neck region of the embryo in this early stage of development. And they ultimately go on to form the cartilage and bones of the face, nose, middle ear, and neck. And defects in the pharyngeal arches, potentially caused by neural crest, can cause some fairly significant facial anomalies and some fairly common ones like cleft lip and palate. And we're going to talk a lot more about those when we cover development of the face. So vagal neural crest cells are not quite as far-reaching as the cranial neural crest cells. They go on to form a portion of the heart called the truncoconal septum, the enteric or gut nervous system, so your gut has a nervous system that controls its movements, and then the thyroid. Now the truncoconal septum is really very interesting. The heart is a complicated organ and it's going to have its own week in class, but just as sort of a quick highlight, at this stage of heart development, the major blood vessels that will arise from the heart, the aorta and the pulmonary artery, are one trunk called the truncus arteriosus. And then they have to divide. And one way they do that is the neural crest cells derived from the vagal region come in and form this truncoconal septum, which in this diagram is also called the aorticopulmonary septum. Same thing. But they form this septum, which allows the aorta and the pulmonary artery to divide into their own separate vessels, which is important, critical actually, for blood flow in the heart. Because if it doesn't happen, you take this is the normal structure highlighted here, and you get this abnormal structure where you have shared blood within the aorta and the pulmonary artery. And if you look at these images, the red blood there being shown on the left is oxygenated and the blue is deoxygenated. So if you don't have this separation, you end up with a mix of oxygenated and deoxygenated blood coming into and out of the heart, which interferes with properly oxygenating tissues and other organ systems. And this heart defect is called truncus arteriosus, which makes sense because that was what the embryonic structure was called. This is a major heart defect and has to be repaired shortly after birth. All right, on to the trunk neural crest. So trunk neural crest goes on to form primarily neurons of various types, although you'll see at the bottom it also goes on to form melanocytes or pigment-producing cells of the trunk and the limbs. There are a couple major neuron structures here that are formed from trunk neural crest, and to talk about them we really ought to just sort of have a quick review of neuron anatomy. So, if you remember, the anatomy of a neuron is composed of a cell body, which contains the nucleus and the genetic information of the cell and most of the cytoplasm, and then the axon, which runs from the cell body to the dendrites. So neurons are meant to transmit signals. So signals that arise in the cell body are transmitted along the axon into the dendrites, which release signaling molecules, which then trigger neighboring cells or the next neuron down the chain to produce some sort of response. So with that in mind, the trunk neural crest cells. So some of them will go on to contribute to peripheral sensory neurons. And in particular, they associate with somites to form these dorsal root ganglia. The dorsal root ganglia are clusters of nerve cell bodies within the spinal cord, the axons then extending to other portions of the body. And so that's these here. We also see trunk neural crest migrating to the sympathetic autonomic peripheral motor neurons, again associating with somites to form chain ganglia. The chain ganglia are also clusters of nerve cell bodies, but they run the length of the spinal cord, and you actually see other chain ganglia next to other major arteries in the body. Now, the sympathetic peripheral motor neurons, those are the ones that control your fight or flight response, so your decision to run or to stay and fight. But the trunk neural crest cells also contribute to your parasympathetic autonomic peripheral motor neurons, which are the ones that innervate your organs and keep things running smoothly without you having to think about it. You don't really want to have to think about making your liver do its thing. Your peripheral autonomic nervous system helps with that. Trunk neural crest also goes to the enteric or the gut neurons. It covers the meningeal coverings of the spinal cord. That helps with sort of protection and buffering of the spinal cord. It contributes to the Schwann cells of the myelin sheaths. Now, the myelin sheaths are the covering that encircles the axon of the neuron. Just sort of go back here real quick. It covers the axon, and that actually helps with efficient transmission of signals. There are many different diseases called demyelinating diseases that result from loss of the myelin sheath. Okay, sacral neural crest cell derivatives. These mostly go to the enteric or the gut nervous system, but very important. So some of you may have heard of Hirschsprung disease. Hirschsprung disease is caused by a failure of neural crest cells to innervate the colon, either all of it or, you hope, if it happens to you, a part of it. And the portion that doesn't receive neural crest cells then can't pulse, and pulsing of the colon moves its contents along towards the rectum. If it doesn't pulse, then contents back up and you end up with constipation of varying degrees, but it can be very, very severe to the point where surgery is required. And some folks with Hirschsprung disease will actually need to have the portion of their colon that is not innervated, so did not receive sacral neural crest, removed as a treatment so that their system can function properly. Finally, with respect to the somites, those clusters of tissue running along the spine, again, some of them are organizing with derivatives of neural crest cells to form these dorsal root ganglia and chain ganglia. They also reorganize in and of themselves into the dermamyotome, which is derived from epithelial cells, forms the skin of the neck and trunk, muscles of the body wall, and limb muscles as well, whereas the sclerotome is more mesenchymal and it will become the vertebrae. Now, we've been talking about the embryo in a very scientific sort of way, but it's worth noting that that's really not how most of the world looks at this. And in particular, what you guys are going to see very rapidly is that the embryonic age that your book is using is not really the same as the gestational age calculation that everything else uses. So embryonic age is the number of weeks post-fertilization. Okay, so from the time of fertilization, how many weeks have occurred? This is how embryologists describe embryos. But everybody else, parents, obstetricians, genetic counselors, most other medical professionals, midwives, use gestational age. And that's the number of weeks since the first day of a woman's last menstrual period. Because back before we knew anything about embryology, all we really had to go on was the thing we knew, which was the last time the woman had a period. And that works out to be 2 weeks more than the time since fertilization. So the first day of your last period is roughly 2 weeks before the day of conception if it happens. So if you're going between the two, embryonic age, okay, is gestational age minus 2 weeks. So if your friend tells you she's 12 weeks pregnant, she's actually 10 embryonic weeks pregnant. Whereas gestational age is the embryonic age plus 2 weeks. So if you're trying to tell your friend about what happens at embryonic week 10, you want to tell her it's actually happening at week 12. And nothing highlights this difference better more than embryologists are thinking about embryos like this, and pregnant ladies are thinking about them like this. If you haven't already, at some point in your life, you will run across the charts that show you the size of your baby based on fruits and vegetables. And for our week, embryonic week 4, gestational week 6, the baby is the size of a pea. Now, from here on out, we're going to be looking at human development from an organ-based platform. So we're not going to go through chronologically anymore. We're going to look at each system individually, although again, with the understanding that all of these things are occurring concurrently.